Hello everyone, let's go over 3 multiple choice questions about microscopes. And in these questions, there's going to be an easy one, a medium one, and a hard one. With that being said, let's just jump right into it. So here's our easy question. The total magnification of a compound microscope is determined by, so remember when we have a microscope, we have something called the ocular lens, which you look through, and then you also have these 3 or 4 sometimes objective lenses. So these are your objective lenses. And what we do to find out the magnification is you multiply them together. So knowing that, let's take a look at our answers. A, dividing the magnification, I can stop there. That's not the answer. It's not dividing, it's multiplying. B, adding the magnifications of the lenses used. Nope, that's also not the answer. C, multiplying the magnifications of the objective and ocular lens. That sounds right, but I'm going to check D and E. Subtracting the objective and ocular lens. Nope. And then E, multiplying the ocular and objective lens by the light intensity. Nope. That started sounding like another good answer, but when you add in the light intensity, that makes it the wrong answer. That's why C is the correct answer. Here's the medium question. A student incorrectly claims that the light microscope can magnify a bacteria enough to see its ribosomes. Why is this wrong? So remember that eukaryotic cells are much larger than prokaryotic cells. This is like a prokaryotic cell. This is a eukaryotic cell. We can easily see eukaryotic cells under a light microscope, and we can also see prokaryotic cells under like the highest magnification of a light microscope, but we can't really see the ribosomes because they're just too small. The reflection of the ribosomes in light just doesn't work where we can actually see them. They're just too small. So knowing that, let's take a look at our answers. A, ribosomes are too small to be seen with the light microscope. That sounds like our answer. Let's check the other ones just to make sure. B, ribosomes move too quickly to capture under a light microscope. No, it's not about the movement of organelles. C, bacteria lack ribosomes. We know that's 100% wrong because all cells have to have ribosomes. So that's just factually wrong. So I'm scratching that one out. D, ribosomes are located in the nucleus. That doesn't sound right and cannot be viewed. No, ribosomes aren't in the nucleus. There are parts of the ribosome that are made in the nucleolus, but they're not actually in the nucleus, so no. E, ribosomes are transparent and reflect no light. No, we really didn't talk about that at all. They're going to have some type of reflection of light, but it's just not enough that we're going to be able to see them. So A is our correct answer. Here's our hard question. Which statement best describes how light travels through a compound microscope? Remember that the light is going to go through our condenser, which is going to kind of focus and condense all of the light. Then it's going to go through our microscope slide and whatever's on it. It's going to go through our objective lens next, where there's 3 of them. It's going to go through our ocular lens, which is at the top, and that's what we look through, and then it's going to enter our eye. So looking at all of these answers, they all start out with light, and they end with the eye, which sounds good. Let's take a look at everything in between. A, light, specimen, condenser. Nope, condenser comes first. Light, condenser, specimen, objective, ocular, eye. That sounds right. Let's check C, D, and E. Light, ocular, specimen, objective, condenser. Nope, not in the right order. Light, condenser, objective, specimen. Nope, comes through the specimen first. And then light, condenser, specimen. Yep, again, no. It is not the ocular first. So the answer of B is the correct one.